Hello, good afternoon, everyone.
Afternoon, sir.
I hope you are doing well.
Yes, please.
That's good. Please, can you see my screen?
Yes, we can.
Thank you. So last week we got to the media preparation and then the types of media. I think we got to the last one, the L and J Lowenstein ginseng medium. So we started with bacterial growth. So once we know the conditions for growing microorganisms, which are temperature, nutrients, pH condition, oxygen requirements, water, or osmotic pressure. Once you provide these conditions, bacteria or microorganisms will grow. So you focus on the growth, and once there is growth, the cells increase in size. So when the cells increase in size, it gets to a point where they mature and they can divide by binary fission. If you are talking about the bacteria, so they divide at every generation, and then two daughter cells arise from the parent cell, and they grow, increase in size, and the two daughter cells, which are now parent cells, will also divide again. So that's how bacteria grow. And once they can grow at a very fast rate, they get into the human host and they can cause infectious diseases. And looking at how they also grow rapidly, we can manipulate them to produce vitamins, to produce insulin, to produce antibiotics for us. Please, is the slide moving?
No, sir.
Okay, let me check. Please, has it moved?
Yes, sir.
Okay, thank you. So once they grow, they multiply by binary fission and in a transverse manner. So they divide along the transverse plane, and the next generation or doubling time will also result in growth of the population. So once you provide all the nutrients they need and you put them in a test tube that has this nutrient, or on the petri dish that has this nutrient, and you don't change the nutrients, assuming they are using the same nutrients for 24 hours, the population size will increase and they will be competing for the same nutrients. As they continue to use the nutrients due to metabolic activities, they will also generate toxic materials. Some of these toxic materials are also harmful to the bacteria themselves. So as a result, some will die, and then there will be competition for the same nutrients. A time will come when the nutrients get depleted, and so there will be nothing for them to live on, and eventually they all die. So once we have a batch culture. Batch culture is when you have the same organisms multiplying and then using the same nutrients. So we don't have the nutrients replenished or changed over time. So that's referred to as a batch culture or a closed system. So there are 4 distinct phases that bacteria will go through or microorganisms will go through when they are in a closed or batch system or a batch culture. So we have the first phase, which is the lag phase, then the log or exponential growth phase. So you see there is a rise in the curve. Then it gets to a stationary phase where it stabilizes. There's no increase nor decrease in the population size. Then it gets to the depth or logarithmic decline phase. Then it comes to 0. So in the lag phase, this is where they are adapting to the environment. Assuming you pick them from a water source or you pick them from a human host, could be blood, specimen, or stool. Once they come out of the human host into a new environment, it will take some time for them to adapt. But if you keep them in the right media, the transport media or in the right media, the lag phase may be very short. But if you keep them at low temperature, the lag phase may also be very long because it will take a while before they come out from the 2 to 8 condition and adapt to the 37 degrees Celsius. So once they adapt, then they will start multiplying constantly. So binary fission is effective in the log or exponential growth phase. The same nutrients are being used, so they will generate toxic materials, they compete for nutrients, so survival of the fittest. Those that are dying and then those that are multiplying, there'll be a balance. So the population size does not increase nor decrease because those that are multiplying are making up for those that are dying, the numbers. So that's why it's referred to as a stationary phase. Then nutrients would begin to deplete and the population size also go down. So if you want to keep the organisms, you change the media. You take some isolates and then put them on a new medium, and this is what's referred to as subculture, so that you don't come to this death or logarithmic decline phase. So you always sustain the growth of these organisms. But if it's in a batch culture where you don't change the media, these are the 4 phases that they will go through. So this is the bacterial growth curve. So I'll explain the lag phase. Usually when you introduce them into a fresh medium or you brought them from the fridge or freezer after storage for some time, then they begin to adapt. Some of them might have been injured and they require some time to recover in the course of storage. And then they will take off to the exponential phase where they begin to multiply. So that's why you require that you keep the specimen in the proper condition. Otherwise, the lag phase may be very long. Assuming it takes 24 hours to produce 1 billion of these bacteria, and the lag phase is long, that means the number of bacteria cells will not be up to 1 billion because of the poor conditions under which they were kept. Or if you bring them from one medium to another medium which has different chemical composition, it will take a while before they adapt. So always take note of what is going on in the lag phase and then how you can shorten that time. In some cases, when the specimen is collected and right away they are put on the required medium, the lag phase may be very short or absent. So it takes a very short time for them to adapt and then move to the log or exponential phase. So this is where you have rapid growth and the organisms will continue to use the nutrients until it gets to the stationary phase where there is cryptic growth. So here, there's a balance between those that are multiplying, dividing actively, and those that are dying. So the population size is stable. Still, there will be some dying as a result of the toxic waste that are produced, example, lactic acids, organic acids from sugar fermentation, and other metabolic activities. But some organisms, once they are in the stationary phase and nutrients become depleted or are depleting, they produce starvation proteins, and this can increase the virulence factor. So, example, Salmonella typhimurium, they can be virulent when they are starved of nutrients for a while. Then the death phase is where there is irreversible loss of the ability to reproduce. So the organisms don't have any nutrients to get energy to go about their metabolic activity. So life ends there. So once you provide the nutrients as a substrate, the concentration of the substrate will reduce as the organisms increase in population. So substrate concentration will decline, and then the population size also increase. It gets to a point where there's no nutrient, and then it comes to zero. So isolation and enumeration of bacteria culture. When you grow the organisms, assuming you pick samples such as blood or stool or sputum, and you provide the right nutrients, the other conditions necessary for growth, you want to assess if there is growth, and then if possible, you want to count. For some of the methods, you may not be able to count the bacteria. For some, you may be able to count, especially if you grow on solid media. Please mute. Diana and Dorcas, please mute. Thank you. So we have direct microscopic count. And here, there are certain chambers that can be used. Example is a Petrov-Horsa counting chamber, the HEMA cytometer, or the Neuber chamber. These are mostly used for counting. They have some grids or some lines on the chamber. So you count the colonies in each small box, then you have a factor which is the volume of that particular chamber. So they have different volumes that they pick. So the size of that chamber, the colonies that you count, then you're able to assess how many you have actually in your sample. Then the variable count using the pour plate or the plate spread technique, where you prepare your media and you pick your specimen, sample of it, and then you place the samples on the petri dish, then you pour your media, then you gently spread the media and then mix it with the specimen. Then you incubate for 18 to 24 hours. Then you come to observe the colonies that are on the medium. Then stability measurement, this is for broth. So for liquid media or the broth, you can assess the growth by looking at the turbidity, the optical density of the medium. So when it's not having any so if there is no bacterial growth, the medium stays clear. But if they begin to grow, the turbidity will result. Sometimes it becomes very turbid, and you can use that one to assess the microbial growth. You can also use the measurement of the cell mass, weight, or volume of the cells by centrifugation, or you can use molecular techniques to measure the proteins or the DNA content. So I'm sure you've been hearing of microbial load using the molecular approach, example PCR. So this is a typical. So Cecilia, the turbidity. Let me show you what the turbidity is about. Do you see this slide? There are two tubes with media, liquid media, broth in them. One is clear, the other is cloudy. So if you put your samples in the liquid medium and there is no growth at that point, it stays clear. When the organisms begin to grow and multiply, the broth will change to become turbid, cloudy. So the optical density will not be the same. And this is how we measure growth in liquid medium. All right. So the variable counting method, you can go about the pour plate technique, or you can use the serial dilation to estimate the variable counts. So you pick your sample or your specimen, then you place it on the petri dish, then you pour your medium, which will supply the nutrients. You allow the medium, before it solidifies, you mix gently, and then solidifies, you incubate, and then you see the colonies. Then you can count the number of colonies on the petri dish. But you have to take into consideration the volume of the sample that you used. Then if you are using the serial dilation approach, here you can go about by preparing your first specimen. So you can have 1 ml of your sample in 9 ml of the broth, the liquid medium. Then you mix gently. Then you pick 1 ml from the resultant solution. Then you plate on petri dish that has a medium, the solid medium, because you're going to count the colonies. But this is the liquid medium. Then you pick another 1 ml of the first one into another tube that has 9 ml of the broth. So the first tube will have 1 ml of the sample in 9 ml of the broth. So that is 1 in 10 dilution. 1 part of the sample and 9 parts of the broth. So that is 1 in 10 dilution. But the second one is 1 in 100 dilution because the first one was a 1 in 10 dilution, and you have picked 1 ml into another tube that has 9 ml of the broth. This broth this time does not have any organism growing in it, but you have picked 1 ml from here and then added here. So this is no longer 1 in 10 dilution. It is 1 in 100 from the previous one. So we do the third one, that is 1 in 1,000 dilution. Same for the fourth one. So this is a dilution factor of 10. Increase by 10 at every step. So you plate from each tube and incubate. When you incubate, the number of colonies you see on this dilution, so 1, 2, 3, 4, the fifth one will be far less than the number of colonies you are going to see on the first one because this is 1 in 100,000 dilution. It's just 1 in 10 dilution. Usually, if you use a 1 in 10 dilution, counting becomes difficult because you have several colonies that are overcrowded. But as you move to the right side, the colonies become distinct. So you can count the colonies for probably this one or that one, and then take into consideration the dilution factor, and then the volume that you use for plating. And this is how you estimate the viable count. And they use this mostly in the food industries, water industries. FDA also uses this to estimate the microbial quality of the food and then these products that are for human consumption. So you can count the colonies, then you know that these are the viable ones in the food or water. So that's the turbidity measurement I was talking about. The first one is having no growth, but if there is growth, it changes to turbid broth. So it becomes cloudy. Another way to also see if the organisms are growing is that when you open this, you have a bad smell compared to the first one that has no growth in it. But you can just look at it from the point of turbidity without opening it. So this comes to bacterial staining. After you have cultured organisms using the right media, you have been able to count some of them. But you then want to conclusively say that this is E. coli, this is Salmonella. So we first need to stain and then have the morphologies, and then that can give you a clue of the type of bacteria. Yes, Millicent.
So please, I want to ask a question on the tips, the turbidity.
All right.
Where you brought the 1 to 5. You said when you are to count, using the first one, the bacteria will be crowded and you'll not be able to count. So which of the levels that will be easy to count, like the best one to use?
So if you count, can you see the cursor, please?
Yes, please.
So if you count from this one, this plate, it's the same as counting from this one, but you have to calculate using the dilution factor. This one is 1 in 10,000, is 1 in 100,000. So if you are seeing less, it's because of the dilution. You have diluted further, that's why you are seeing less. But if you calculate using the dilution factor, it will be the same as what you have here. So you can use any, provided you can see distinct colonies. If they're overcrowded, it's not good to use that one to estimate. That's why we do dilution and then able to see the distinct colonies and then use that one. So if you are seeing this clearly and the colonies are distinct, you can go for this and not the last one. Is it okay, Millicent?
Yes, sir. Thank you. So the fourth one will be effective.
Yes. So I would go for this one and not the first three because they are overcrowded.
No,
So we have a question. Is it the same count method used for calculating for MPs? You mean malaria parasites? Are you talking about the malaria parasites, Plasmodium species?
yes, sir.
So this one, the estimation is different. This is for bacteria. So for malaria parasites, you count the number of infected red blood cells. If you are using the thick film, so we have the thick and the thin film. So you count, if you are using the thick film, you count the number of infected red blood cells that will have the parasite, and then the number of white blood cells. So the parasite that you count per the white blood cells, and this is different from the serial dilution. Is it clear, please? So for the malaria parasite count, you don't dilute, you just use the whole blood. You count the number of parasites in the fields, and then you estimate per the WBCs. If you go to the thin film, then you count with the RBCs, per RBCs. Right. So bacterial staining, if you pick the raw colonies and then you want to visualize under the microscope in a solution, which is aqueous, after emulsification, they will appear colorless because the bacterial cells have almost the same refractive index as that of water. I hope you understand refractive index. Hello?
No, please.
So when we talk of refraction is how light changes direction when it enters a new medium. So assuming you throw light from air, it enters water. Water and air are not the same in density. So it will change or bend. That is refraction. That's why you put a pencil or a pen in a bottle of water, and then from the outer part, you may see it as a broken pen or pencil because of the change in medium. So that is refraction. So when light passes through bacteria, the degree of refraction is almost the same as that of light passing through water. And because they have almost the same refractive index, which is the measure of the density of that particular medium, you cannot see the bacterial cells when they are suspended in the aqueous solution. Is it clear?
So please come again with that part. Thank you.
So when you pass light through air and then water, we have bending of light. That is refraction. The refractive index is the density of that medium that makes the light bend. So the degree of bending. If you pass light through air and then water, the degree of bending will be the same, almost the same as passing light through bacteria. Because of that, when you have the bacterial cells suspended in aqueous solution, which is water, because they have almost the same refractive index, you may not distinguish the bacterial cells from the water. That's why we stain the bacterial cells or impart color, and then we can then see the bacterial cells in the midst of the aqueous medium. Is it clear? All right. So this is the purpose of staining. You want to impart color, and then we can see that against a background. So we use what you refer to as biological stain. And mostly in hospitals or research facilities, they make use of the light microscopy. There are others, the fluorescence and then the electron, but for the purpose of diagnostics, we just want to see whether the organisms are there or not. So, and then count. You're not interested in the fine details, whether it has chromosomes, whether it has nucleus or not. You want to see whether it is there or not. So that's why the light microscope is employed. But for research purpose, they want to look at the fine details and they go for the very powerful ones, the electron microscope. So when you talk of a biological stain or dye, it has three components. It has an organic solvent containing a benzene ring. It has a chromophore that imparts color to the organic compound. Then an oxochrome, which makes it ionized, gives it a charge. So this biological stain will not be able to impart color to the bacterial cells unless it has a charge. And this is coming from the oxochrome. I've explained why we stain. Sometimes you want to also look at the different internal structures of the organism. So when you stain with specific dyes, be able to increase the visibility of those structures. And also stain to preserve them for future use. And before you stain, you prepare your smear. So after growing the bacteria on the media, so you can use the liquid or the solid medium. If you are using the liquid medium, there's no need to resuspend again in water or in saline. But if it is on a solid medium, like the agar, you pick a colony and then you emulsify in normal saline or water. Then you apply it in a circular motion on the slide, which is cleaned and then air dried. If there is oil or debris on the slide, you're going to see artifacts. So always make sure the slides are clean. Then you prepare your smear, you label accordingly. And it shouldn't be too opaque because remember, you're going to use the light microscope to observe. So light should be able to pass through the slide and then get into the objective lens. So from the light source, it goes through the condenser lens, the objective lens, it goes to the ocular lens before you can see the virtual image. But if this is too opaque, light cannot pass through, and this is going to give you a negative outcome. Please mute. Juliet. So this is what we refer to as a smear. The resultant solution that you have placed on the slide for staining. So for bacterial staining, you air dry the smear and it will appear translucent. So you can view this against light and then check if you can see through. So if it's translucent, that is okay for staining and then observation. Then you heat fix the smear. Once you heat fix, it enables the smear to adhere firmly on the slide because you're going to stain and then wash in between. If it is not fixed, it's going to get washed off the slide and then there will be nothing to see. Once you apply heat also, it kills the organism. So this smear no longer becomes infectious in some way, cannot be a source of infection. And it also allows the smear to pick up the stain readily. So you prepare your glass, you clean it, you dry any grease or oil you have on it, make sure they are off, you label accordingly, prepare the smear, avoiding the thick and dense smear, and then you can then apply your sample. So it can be from a liquid medium, broth culture. So if it's blood culture, you can directly use the blood culture, and then there's no need to emulsify in saline or distilled water because it's already in the liquid state. Or if you incubated in any liquid medium, but if it's from a solid medium, then you pick some colonies that are distinct, and then you emulsify in saline or distilled water, and then you prepare your smear. Then you heat fix, as I've said earlier, to have the proteins coagulated, and then stain on the glass so that they don't get washed away during staining and washing. So you can heat fix by passing the slide, which has a smear on the benzene burner, a flame, or so Winfred is asking what's included in the labeling. I can see some numbers. Okay, so if it's hospital based, you can use a hospital ID. Or if it's not for research purpose, you can use the patient's name. But for research purpose, you anonymize, you code them. So this can refer to the hospital ID for that particular patient. All right. So you hit fix. You can use a benzene burner, which will produce a flame, and then you pass it 2 or 3 times over the flame gently, not in a fast manner. Or you can place it on a micro incinerator, or you can have it on a hot plate. So the micro incinerator, you can just move it in front and not have the smear destroyed. But if you can regulate, this time we have the automated type, you can regulate and then you can just place it in, or you can have it in the dry air oven, but you regulate the temperature. So the biological stain has the benzene, in the organic solvent, which is always colorless. And then the chromophore, which gives the organic solvent its color. So the organic solvent, which contains the benzene ring plus the chromophore, is referred to as chromogen. So chromogen is just a colored organic solvent, but it is not yet a stain because it's not charged. So the ozochrome will then ionize the chromogen. The chromogen is the organic solvent and then the chromophore. Once the ozochrome is added to the chromogen, then it is a biological stain or dye. And this can be used to stain the cells and the tissues. And then impart color. You can distinguish them from the background and then use it for your diagnostic or research purpose. And then you can differentiate different types of organisms based on the colors that they exhibit. You can also look at different parts of the organism if you are using a powerful microscope. So for the biological stain to bind depends on the charge on that organism and then the charge that the chromogen is also carrying based on the ozochrome portion. So if the organism has on its external part negative charge, and you are using a stain that is also negatively charged, there will be no binding because negative, they repel. It also depends on the cellular components to be stained. So any stain that you buy, they indicate the composition. And then from there, you can know the charge on the chromogen portion. So we can classify the biological stains based on the charge when they are ionized. The ionization is coming from the ozochrome. Take note. The chromogen is the organic solvent which contains the benzene plus the chromophore. You can also classify the stains, the biological stains, based on the function, what they are used for. So you can use this to differentiate one organism from the other, or you can just use it to impart color to all cells. So for the charge, you can have acidic and then basic biological stains. So stains that contain sodium, potassium, calcium, ammonium salts, they are negatively charged. So the chromogen is negatively charged and they are the acidic stains. For stains that have the chromogen portion being positively charged, they contain the chloride or the sulfate salts. So based on charge, basic, acidic, or neutral. Neutral is when you have those two combined. So a stain with a positive charge coming from the ozochrome is a basic stain or basic dye. A stain with a negative charge is an acidic stain or acidic dye. So bacterial cells are slightly negatively charged, and therefore, which one would you go for? Basic, because negative binds to positive. If you go and use an acidic stain, it will not impart color to the bacterial cells. to rather stain the background. So basic stains are what we use to stain cells surfaces that are negatively charged. To stain positively charged molecules of the bacterial cells, the individual components or structures, then you can go in for the acidic stain. And for this, you need to use the powerful microscope to see the individual structures that are stained. For example, the capsules, and then the background also gets stained and not the bacteria. So acidic stains are also referred to as anionic stains, and they have negative charge coming from the oxochrome to the chromogen part, the colored part of the organic solvent, and then the chromophore. And because they are negatively charged, they have very strong affinity for structures that are having positive charges. So using these anionic or acidic stains will bind to these positively charged components. Examples of acidic stains include picric acid or yellow stain, eosin, Indian ink, nigrosin. Examples of basic stains or cationic stains are methylene blue, safranin, crystal violet, malachite green. I'm sure you are familiar with the malachite green from the L&J medium. So basic stains exhibit positive charge, and because they are having a positive charge, they have strong affinity for negative constituents of the cell. And this is what we use to stain bacterial cells. Somebody is talking about xylene. Xylene is not a stain, please. So is there any difference between stains and dyes? So a dye can stain technically anything, but the technical word is stain. So a dye can impart color to anything. But when you talk of biological stain, it should have the three components. And it's staining based on the charge that is carrying in that particular that particular biological stain or the biological dye. So in this case, biological dye, because it's going to stain the cells or the tissues. And it also depends on the charge on that particular part of the cell or the tissue. But a dye can impart color to anything without a charge. Simon, do you get the difference?
Yes, please.
Right.
Yes, sir.
So Raja is asking, does this staining being negative or positive charge have any effect on whether the bacteria is So that's what we are able to use to distinguish bacteria as being gram-positive or gram-negative. We shall come to but it's not the charge on the chromogen that is making the bacteria gram-negative or gram-positive. Raja, please take note of that. Bacterial cells are, the external part are slightly negative. That's why we use the basic stain. This has a positive charge, so it will bind to the slightly negative external part of the bacteria, which is the cell wall. But when you talk of gram-positive, gram-negative, we shall come to why they are called positive and negative. But it has nothing to do with the charge on the bacteria structure. The external part is always slightly negative. Is it clear, Elijah? All right. So negative, where are we? Yes. Okay. Negative staining. Here, you want to study the morphology, the size, the arrangement of the bacterial cells. So please take note of this one. Negative staining is when you stain the background and not the bacteria. So in this case, you have an unstained bacterial cell, but a stained background. So this will reveal the actual shape and then the size of the organism and then the arrangement. But the actual bacterial cells are not stained. So if the bacterial cells are slightly negative, and then use an acidic stain which is also negative, then you are not going to stain the cells. And therefore, the background will get stained or colored, and the cells remain unstained. But if the cells are not stained and the background is stained, you can still see the morphology. So this is how you see it. The background is stained, but these are the cells. They remain unstained, and you can see the shape, the arrangement. And this is what we refer to as negative staining. The background is stained, but the cells are not stained. So you use a stain that has the same charge on the bacterial cell, and therefore the background remains stained. For negative staining, because you are not interested in staining the cells, the bacterial cells, you don't heat fix. Because you're not interested in making the cells adhere to the stain. You just want to stain the background. So we come to the staining based on function, classification. So we can classify based on the charge or the function. Please mute. Frances. So based on function, take note of this one. We have simple staining and differential staining. The difference is in simple staining, only one stain is used. Differential staining, you can use more than one stain. You use more than one because you want to differentiate some bacteria from others. Simple stain, because you use only one stain, all the cells, whether it's mixed infection or not, all the cells from all the isolates will pick the same color. So you're not able to differentiate. But if you use different stains, that becomes differential. You're able to distinguish gram positives from gram negatives or acid fast from non-acid fast bacteria. All right, so simple staining, you use a single stain. Differential, use two stains that do not have the same color. So in some facilities, this is mostly done in emergency care when the clinicians need results as soon as possible to initiate treatment. The lab person may go ahead to use a simple staining just to see the morphology. So are they cocci? Are they bacilli? Then the clinician may have an idea of the organism to start treating because this is emergency case. Or if it's about looking at the morphology or the arrangement without necessarily looking at what type of bacteria they are, then you go for the simple staining. It saves cost anyway, but it has some disadvantages because you cannot differentiate if you have mixed colonies in the specimen. Differential can separate them into groups based on the different colors that they exhibit after staining. You can also visualize structures such as the flagella, capsules, spores. So if you use particular stain for the external part, you can also use specific stains for the flagella and the other parts. Please mute. Christiana. All right. So special staining is when you want to stain these structures, capsules, spores, flagella, metachromic granules, and so on and so forth. So these granules are nutrients that have been accumulated within the bacterial cells, and they can be used to also, yes, who is this? The name appears as Techno. Yes, you have a question? All right. So simple staining, only one stain is applied. So you can use crystal violet, methylene blue, basic fusion, malachite green. So if you're able to use all these stains on bacteria, then are they acidic or alkaline basic stains? And what is the color, the charge, sorry, exhibited by the chromogen? I hear Lydia, your video is on. Kindly put it off. So the question is crystal violet, methylene blue, basic fusion, or carbon fusion, malachite green. If these stains, these biological stains can be applied on bacterial cells, then are they acidic or basic stains? Is the question clear, please? So we have two answers, acidic and basic. Which is which? So let's go back. So you see them here, methylene blue, safranin, crystal violet, malachite green. They are basic because they exhibit positive charge, and this is going to bind to the slightly negative charge outer part of the bacterial cells, right? So the one who said acidic, that means you are going to stain the background only. All right, so once you apply only crystal violet, that becomes simple stain because you are using only one stain. If you apply only methylene blue, only basic fusion, only malachite green, becomes simple staining. So it gives color to all the bacterial cells, then you are not able to differentiate because it's the same color that is applied to all. You can only see the morphologies if they are spherical, if they are bacilli, if they are comma shaped. You can see the arrangement if they are in pairs, in clusters, or in chains. So you prepare your smear as earlier said. You heat fix because you want to stain the bacterial cells. This is non-negative staining, so you heat fix, then you apply the basic stain, and then you allow for about 1 minute. Then you wash with water gently so that you don't wash the smear off. You air dry, then you focus with the microscope, and then you record. So for microscopy, you focus with the low objective first, and then you change to the high objective. You don't start right from the ×100. And if you are using ×100, you have to use immersion oil. I hope you know why immersion oil is used. All right, let's move on. So once you apply a simple stain, okay, so for the microscope, the light microscope, ×10, ×40, those are the low objectives, ×20. It means that this one can magnify your object size 10 times. ×40 means it can magnify the object size 40 times. So it produces a virtual image 40 times. ×100, same. And for the ×100, because the image is going to be enlarged, we are likely to have issues because there can be diffusion of the light rays due to the distance between the lens and the object. So in this case, what is on the slide. So you place an oil on the specimen, and this fills the gap between the lens and the object, which is on the slide. Remember, when light changes its path from one medium to the other, it bends, doesn't travel in a straight course again. So if the light enters the slide and there is space between the slide and then the lens, you're likely to have the light rays bending again, and then this may diffuse. But the oil will have almost the same refractive index as the glass, and then this makes the light rays focused onto the objective lens. So that's why we use the immersion oil for the ×100. So if you do the simple staining, you have only morphologies seen, but you cannot differentiate one from the other. You can also see the arrangements. So prepare your smear, you dry, heat fix by passing over the flame, or you can use a hot plate. Then you apply only one stain. Then you allow for 1 minute. Then you wash, you rinse, you air dry, and you observe under the microscope. So this is where you place the oil. This is your slide, which has your smear stained and then the oil is placed here. Then it fills the gap between the glass and then the objective lens times 100 objective lens. So I've thought about the simple staining and it's very rapid, saves cost and time. But the disadvantage is that you cannot differentiate some organisms from the other. It gives limited information because if there is mixed infection, then you cannot classify which type of bacteria you should treat. That's why we're going for the differential staining in most health facilities. Here you use 2 or more dyes of different colors, so biological dyes or stains, and then you can apply them sequentially. In between, you wash, you decolorize, and then you're able to detect the one which picked the first or the second biological stain. This one gives more information because you can differentiate based on the different colors that they exhibit. So there are 2 main types of differential stains in bacteriology. So for bacteria studies, we have the gram stain and then acid-fast stain. So you can use this gram staining technique, which was developed by Hans Christian Gram, to differentiate gram-positive bacteria from gram-negative bacteria. And it relies on the differences in the bacterial cell wall. And some antibiotics also target the cell wall anyway, not on all antibiotics. So if the bacterial cell wall is very thick in what we call peptidoglycan, I think we did this in the previous lectures, bacterial cell wall. Do you recall? Peptidoglycan. Hello.
Hi. Yes, sir.
Peptidoglycan, yes. So we have the peptidoglycan in the bacterial cell wall. But some are very thick, some are very thin. Those that are very thick are the gram-positive bacteria. Those that have very thin peptidoglycan layers in the cell wall are referred to as gram-negative. So here you see that it has nothing to do with charge. The one who has a first question. Gram-positive, gram-negative, it has nothing to do with the charge. They are both negatively charged bacteria on the outer part, but the gram-positive bacteria have very thick peptidoglycan layers. Gram-negatives have very thin peptidoglycan layers. And because one is thick, one is thin, they will exhibit different colors when you apply the stain sequentially. We shall go through that. So gram-positives will keep the first stain, which is the purple color. Gram-negatives will lose the first stain when you decolorize. And then they will pick the second stain, which is the pink. That's how you're able to see gram-positives and then differentiate them from gram-negatives. All right. So for gram staining, these are the reagents that we use and then why they are used. We have the primary stain, which is crystal violet. So this primary stain will impart color to all the bacteria that are on the slide or on the smear. So at this point, it is simple staining, but we want to add more colors or more stains. So this is differential. But at this first point, it is simple staining. Then we come to add a mordant. A mordant in the form of iodine intensifies the color of the primary stain. So it forms a complex between the primary stain and the iodine so that they can bind readily to the cell wall. Then decolorizing agent, you try to remove the stain from the first one that you applied. And because gram positives have very thick peptidoglycan layers, they will keep the first stain, the color. Because gram negatives have very thin peptidoglycan layers, they will lose their primary color. So at this point, when you decolorize, you have one being purple, one being colorless. It's just like you are washing towel and then handkerchief. Both are stained. So the towel is analogous to the gram-positive cell, which has very thick peptidoglycan layer. Then the handkerchief is analogous to the gram-negative cell, very thin peptidoglycan layer. So when you are washing the two, the handkerchief will lose the stain easily than the towel because they don't have the same thickness. So once you apply another stain, the one that remained colorless, which is the gram-negative, the one that lost the primary stain will then pick the contrasting color of the counter stain. But because the primary stain is still in the thick peptidoglycan layer, the gram-positive bacterial cells, they will not pick another contrasting color. So we have the first one keeping the primary stain, then the one that lost during decolorization picking the second stain. So that's how can be able to differentiate gram-positive bacteria from gram-negative bacteria. So we decolorize with acetone or ethyl alcohol, and the counter stain is usually carbon fusion or saffron. So crystal violet is a primary stain, which is violet in color or purple. The grams iodine or the logos iodine as a mordant to intensify the color imparted by the primary stain, the decolorizer to remove the stain, and the counter stain to impart color to the cell or that lost the primary stain in the course of decolorization. So it is comparison between gram-positive. So we see plus here, negative. So gram-positive cell wall, very thick peptidoglycan layer. Gram-negative cell wall, very thin peptidoglycan layer. So the principle is what I've just explained. So the principle is that gram-positive bacteria contain high cross-linked peptidoglycan layers. On the other hand, gram-negative bacteria contain very thin peptidoglycan layers. But if you apply a primary stain or a primary biological dye, such as crystal violet, all the two will pick the same color. They will become purple or violet. If you add the iodine, it will intensify the color imparted by the crystal violet to both. And this will form the iodine crystal violet complex in the peptidoglycan. Then you try to remove the color imparted by the crystal violet, the primary stain. So you add a decolorizer. Because this is very thick, it will retain the primary color within the time that you have allowed. So within 5 seconds, the crystal violet will not be removed entirely from this peptidoglycan, but within 5 seconds, this thin peptidoglycan layer will lose the primary stain. So this will become colorless. This will keep the purple color. Because this has become colorless, you then apply a counter stain, which is safranin or carbon fusion. And this will make the thin peptidoglycan layers take the counter stain and then become pink. So you wash with water. Water is not a decolorizer. So tap water will only remove excess stain on the smear. You dry and then you observe under the microscope. So that's the principle. Is it clear, please? Simon?
Yes, please.
Okay. So principle is what I've just given you. But if you are asked to just go through the steps, you add the crystal violet, you time for about 1 minute, you add the grams iodine, you time for about 1 minute, you decolorize for about 5 seconds, 3 to 5 seconds. So when you see color coming out, then you wash gently with tap water. Then you apply the counter stain. You will time for about 1 minute. So it depends on the protocol. Some time for 5 seconds, time for 1 minute. Please mute. So those are the steps. By principle, you have to add the reasoning. So when you apply the primary stain, the mordant to intensify the color. So you see it has become very deep compared to this one. Then you decolorize. Some will lose the primary stain, some will retain. So those that lost the primary stain are the gram negatives. Those that retain the primary stain are the gram positives. Then you add the counter stain, which will then color the ones that lost the primary stain in the course of decolorization. So this is the purpose of gram staining. So gram negatives are pink, gram positives are purple. But you add the reagents sequentially. So follow the protocol and then you are good to go. So this is what I've just explained. Then you observe under the microscope, starting from the lowest objective, then you focus and then you observe critically with the highest objective. So more illustrations for you. crystal violet, iodine, alcohol or ethyl alcohol, acetone, saffron, or carbofishing. Then we talk about the next differential stain, which is acid fast or Ziehl-Neelsen staining. There are some bacteria that you cannot apply the gram staining method to because they resist decolorization, this step. Please, can you see the cursor?
Yes, sir.
So Winfred is asking, what's the purpose of alcohol? So the alcohol is to remove the stain. It is an organic solvent. So once you add alcohol, it removes the stain. That's how the gram negatives are able to lose their primary stain. But because of the short time, the alcohol will not be able to remove all the stains in the gram positives. So here you do it briefly within 5 seconds. Those that are thin peptidoglycan layers will lose the primary stain due to the alcohol application. Winfred, is it okay? So alcohol will not kill. So that's why we allow. Oh, okay, I understand your point now. The alcohol we are applying here, already we have heat fixed, so the bacteria cells are dead. Take note of this one. Here you want to impart color. We have already heat fixed to kill the bacteria already. The purpose is to impart color. So when you apply the stain, the alcohol is to remove the stain. Winfred, is it okay now? Before we move on, are you okay? All right, good. So you move on. Was there a question earlier? Let me check. Right. So we are on Ziehl-Neelsen or acid fast staining. Yeah, so on the acid fast, I was saying that there are some bacteria that will resist decolorization with the usual ethyl alcohol. So if they are able to resist this decolorization, then it will be difficult to know if they would pick or retain the primary stain. In that case, we don't use the gram staining technique for those particular bacteria because they are acid fast. They resist the decolorization. They hold fast to the decolorization step. And most of these types of bacteria are the mycobacteria and nocardia. So nocardia, unlike the mycobacteria, they have less of the lipids that prevent this decolorization, but the mycobacteria, they have a lot of these lipids, the mycolic acids, that retain or resist the decolorization. So we term them as acid fast bacteria, AFB. They have high lipid content. So Yakubu is asking, if you over decolorize, this is what will happen. If you decolorize for a long time, assuming you extend to 10 seconds, it means that with time, the gram-positives will also lose the primary color. Just like you want to remove the stains from the towel and then the handkerchief, you are using bleach. If you allow a short time, the handkerchief will lose the stain compared to the towel. But if the towel stays in the bleach for a long time, it will also lose the stain. That's why we allow less time to decolorize. Yakubu, are you okay? So if you over decolorize, you are going to see all of them as negatives. That's the effect. And that can affect the clinician's decision to start treatment. Meanwhile, the antibiotics he or she may be prescribing will be for gram-positive, but because you have over decolorized, you'll be reporting as gram-negative. And sometimes it happens in the lab. If you have the lab people working on so many things at the same time, they may forget. And then once they come back, they see the smears over decolorized. And instead of repeating, using another isolate, preparing the smear again, some of them, due to laziness, they will just ignore and then go ahead to wash and then observe. Okay, so we use the acid-fast staining or the Zell-Nelson staining. So two scientists came up with this, first by Zell and then later modified by Nelson. These organisms are not able to take or undergo the usual gram staining because they resist decolorization. So they are termed as acid-fast. And therefore, we use the Zell-Nelson technique. So here, we also use carbofishing, which is a basic stain, combined with phenol. Phenol is a lipid solvent. So once you apply this carbofishing with phenol, it makes the carbofishing readily impart the color to the bacterial cells that have very high glycolic acid content. So with the presence of the phenol, it binds to stain the cell wall, which has this glycolic acid in the mycobacterium, and then an acid alcohol is applied. Remember, we said that they resist decolorization. So this will remove the background cells and any organism that does not have this glycolic acid. But the ones that have high glycolic acid content, example, the mycobacterium or the nocardia, they will hold fast to the carbofishing that you applied first. That's why we say they are acid fast. While the other organisms, the other bacteria that are not having the glycolic acid will lose the primary stain, the carbofishing, when you decolorize. And then you add a counter stain, which is malachite green or methylene blue. And this will stain the background and any organism that had the carbofishing removed in the course of decolorizing. But the mycobacteria that have high glycolic acid content will retain the carbofishing. They will hold fast to the carbofishing. That's why they are termed as acid fast. So primary stain, carbofishing with phenol, then sulfuric acid or hydrochloric acid in different concentrations. So for nocardia, because they are weak acid fast bacteria, you reduce the sulfuric acid content or concentration, and then you're able to remove be able to remove the other stains from the ones that are not acid fast. If you use high sulfuric acid concentration for the nocardia, they are likely to also lose the primary stain because they are weak acid fast bacteria. So you decolorize with the alcohol, just like was done in gram staining, and then the non-acid fast bacteria will lose the primary stain, but the mycobacterium will keep the primary stain. But when you apply the counter stain, which is the methylene blue or the malachite green, the mycobacterium will not pick it again because they've already retained or they've held fast to the carbon fission. Those that lost the first stain will pick the counter stain. So the principle is what I've explained, same as the procedure, and you observe. So this time you'll be seeing pink bacilli. These pink bacilli are not gram negatives. Take note, this is acid fast staining. So if you go to the lab and then they show you some slides under the microscope, the first question to ask is it gram staining or acid fast staining? If it is acid fast staining and you are seeing pink, if you are seeing pink, what does it tell you? Hello? If it's acid fast staining and you are seeing pink, what does it tell you?
Gram negative.
Acid fast. Acid fast. This is not gram staining. For gram staining, pink is gram negative. But for acid fast,
The presence of
sorry?
yeah, microbiology.
Yeah, so that's why I'm saying that once you go to the lab and then they show you the pink bacteria, the first question you should ask them is it gram staining or acid fast staining? Acid fast staining is the same as gel-nersine staining. So don't just look at the color and conclude that it is gram negative. You may be mistaken. I hope it's clear.
Yes, please.
So you are seeing the pink because you had carbon fission. Let's go back. Mixed with phenol. Then those that lost the primary stain but picked the counter stain, which is the malachite green or the methylene blue, will have the blue background and then the blue stains. All right. So Elijah, where should I come again? Where are you confused? Okay, so I asked about seeing pink bacilli. If you see pink bacilli, what comes into mind? So what will come into mind will depend on the type of staining. Is it gram negative staining? If it's gram negative, gram staining, pink is gram negative. But if it's about acid fast bacilli, pink is for acid fast staining. So the mycobacteria are going to be pink. They will hold fast to the primary stain. And subsequently, the blue ones will be the non-acid fast bacteria because they will lose the primary stain when you decolorize. So pink is not always indicative of gram-negatives. Depends on the staining technique. Are you okay?
Sir, please. So that means the green malachite or the blue no is the non-acid fast, and the
Exactly.
pink is the acid fast.
Yes. So they would pick up the counter stain because they lost the primary stain when you decolorized. This point. So if you use malachite green, then you're going to see green bacteria cells. If you use a methylene blue, you're going to see blue bacteria cells. And if the microbacterium is present, you see pink. I hope it's clear. All right, so that brings us to the end of culture
Sir,
media.
please, what about the red color?
Which one?
The red color. I saw
This
red
one?
color is acid fast. The red color.
Which
On the
slide? This
next
one?
slide.
This one?
No, no, please. One of the slide I saw red.
Red.
Yeah,
Let me
and
go back.
you wrote acid.
Not seeing that one. Let me check again. Is it this one? This
Yes,
one?
please. Yes, please.
Yeah, so people see pink as red. Even for purple, some say purple, some say um what color? Violet. So sorry,
Mode.
both. So in some books you see red or pink. That's why I've put the red here. But pink is commonly used in books. Thank you. So AFB is acid fast bacilli, and those are the ones that have high mycolic acid in the cell wall. And these ones, gram staining is not applicable. You have to come and use the zeolnecin staining or the acid fast staining. Any question, please? Any question? So we will start a new topic next week, God willing. If you have any question, hello.
The slides.
Oh, you have the slide, don't you?
So this one we are not having.
Oh, okay. I'll send them right away.
Okay, sir. Thank you.
So I think you have up to um, stability, if I recall, or you have up to the media.
We have lecture 2, part 2.
Okay, probably you have up to LNJ. Maybe you don't have this one, bacterial growth. I'll check and send
Yeah,
them to
we
you.
did. Okay.
Okay. Any other question? All right. So Salome, I'll send them to you. So in the absence of any question, I think we can end here. Enjoy the rest of your weekend.
Goodbye, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.